Great. Okay, good afternoon, everyone. Welcome back to our NT-ENDURE virtual seminar series. And today we have the pleasure of having Dr. Elva Díaz. Dr. Elva Díaz is professor in the Department of Pharmacology and chair of the Neuroscience Graduate Program at UC, University of California, Davis School of Medicine. Dr. Díaz has a broad background in biochemistry, molecular and cell biology, and genomics in studies of the mammalian nervous system. And she was born and raised in San Jose, California, to parents originally from Mexico. Dr. Díaz's interest in science and math in high school eventually led her to a career in biomedical research. She earned her bachelor's degree from Harvard in biochemical sciences in 1993, followed by a summer internship at Merck company. And later, she continued to do her PhD at Stanford, where she earned her doctoral degree in 1999. After her graduate studies, in particular, her graduate studies involved the molecular mechanisms of receptor trafficking in non-neuronal cells. And as a postdoctoral scholar, Dr. Elva Díaz switched fields into developmental neurobiology and worked on functional genomics of nervous system developed at UC Berkeley and UCSF. And since 2003, she's been part of the faculty at UC Davis, and most recently, she was appointed chair of the Neuroscience Program. And today we'll have the pleasure of having her talk about her research. And also, after her presentation, she'll be joined by Dr. Martin Husrey, who is professor and chair of the Department of Neurobiology and Physiology and Behavior, and the Barbara Horowitz and John Horowitz Endowed Chair in Physiology at UC Davis. And joined to Dr. Husrey, he'll have one of the graduate students from the Neuroscience Program, Eden Barragan, and the academic coordinator of his T32 grant, Ashley Huddle, that both three of them will be talking about the wonderful program that they have at UC Davis. So without further ado, thank you, Elva, for being here. It's a pleasure having you, and let's listen to wonderful science. Thank you.
Great. Thank you, Carmen, for that wonderful introduction. And it is my pleasure to be able to talk to you all today. I think the idea is that questions will be in the chat, and then we can answer at the end. But that's my understanding. So I will give you an overview of the research that goes on in my laboratory, a little bit about my background and how I got here, and then hand it over to Dr. Usry to talk about the neuroscience opportunities in a little more detail here. And it's meant to be very open and flexible, so please feel free to ask anything that comes to mind during this presentation. So this is a picture of a coronal section of a mouse brain taken from the Allen Brain Atlas, which is a great resource available for molecular profiling of the nervous system. And as Carmen was mentioning, my background was in biochemistry, cell, and molecular in non-neuronal cells, but as a student, I think I was a first-year graduate student. I had the opportunity to attend a Gordon conference called Cell Biology of the Neuron. And I remember being fascinated by the complexity of neurons and the complexity of the nervous system. And so this slide is meant to illustrate the complexity that exists within our brain. So as you see in this coronal section, the different colors are in situ hybridization patterns of different genes that are expressed in different places throughout the nervous system. So you see in red and kind of like pink and green, two genes that are expressed in the cerebral cortex. You see in the cyan color a gene that is expressed in the dentate gyrus of the hippocampus. And you also see in green genes that are expressed in the striatal regions of the nervous system. And so I hope you can appreciate the vast complexity that exists. So also when I was a student, this was at the forefront of the genomic revolution. Now we all take advantage, or take for granted the vast majority of the vast amounts of information that's available to us online in various databases about gene expression profiles that exist in many different organisms as well as tissues. And when I was a student, this was right at the beginning of genomics. And so I apologize, that's probably my email. Let me turn that off. Well, that was right at the beginning of genomics. And I remember being fascinated by the numbers of genes that were expressed in the nervous system. So we now recognize that there's roughly 23,000 genes within the mammalian brain, or within mammalian species, 23,000 within us. And it turns out that at least half of them are expressed almost exclusively in the nervous system, and up to 80% of all genes in our genome are expressed somewhere in the nervous system. So this indicates that the vast majority of our genome, which makes us human, is dedicated to the complexity that exists within our central nervous system. And this is what really made a profound impact on me as I was choosing the area to focus on as a postdoctoral scholar, because I was really excited about the complexity of the system and being able to try to address this in a molecular mechanistic fashion. And for those of you, I know you all are biology majors. You might be thinking like, well, you know, I hadn't necessarily considered a career in neuroscience, but my training is not in neuroscience either, as was illustrated by Carmen. My background's in molecular and cell biology, genetics, and biochemistry. And then this is what we do to try to understand questions of the nervous system. We use all of these different approaches to understand nervous system development and function. And then we collaborate with individuals to do physiology and behavior. And so I want to illustrate that the neuroscience field, you can apply a huge number of different approaches depending on what your main interest is. And it's a fascinating subject, so I would strongly encourage you all to consider it for your future career choices. So for the rest of today's talk, I'm going to tell you a little bit about one of the projects that has been ongoing in my lab since establishing it in 2003. And we focus on a particular area of the brain called the hippocampus, which is outlined here in this white box. That's important for learning and memory. So it's important for how we are able to retain information and recall it at a later stage. And I hope that after this lecture, some of your synapses will be strengthened such that you can recall this information tomorrow, hopefully. So if we were to zoom in on an area of the hippocampus and look to see what these cells look like, you might see something like this. And as I mentioned, this is an example of one of the cells that I saw as a first-year graduate student at the Cell Biology of the Neuron Gordon Conference. And I was just fascinated that this was a single cell, right? So you have the nucleus that has the chromosomes that encodes all the genes that make the proteins necessary for this one cell. In red are the dendrites that are labeled by MAP2. And then in green are the postsynaptic structures called dendritic spines. And you can see that there's quite a large number of these synapses associated with a single cell. And on average, it's thought that each neuron makes roughly 1,000 synaptic connections with other cells. And it's thought that there are roughly 100 billion cells, neurons within our nervous system. And so you can imagine the vast complexity in terms of synaptic connections that can be formed within our brain. And it's thought that this vast complexity is what allows us to have achieved the higher order cognitive function that we rely on our everyday life. Okay. So again, focusing in on the individual fundamental unit of the neuron, which is the fundamental unit of the brain. And I am interested in the molecular mechanisms of excitatory synapse development and plasticity. So if we were to zoom in on one of these synapses and then convert it to a cartoon, this is what it might look like. So some of you are likely familiar with this structure. So this is a classic excitatory synapse, whereby you have the presynaptic input on the axon, which has the synaptic vesicles filled with neurotransmitter, and these are docked at the active zone. This structure, this presynaptic input, forms a specific and tight connection with the postsynaptic dendritic spine, which is shown here, through the adhesive, transsynaptic adhesive complexes that form. And then on the postsynaptic side, you have the receptors for the neurotransmitters. In this excitatory synapse, there are NMDA receptors and AMPA receptors, and these are held very tightly in this structure at the postsynaptic density. And so what I am interested in is understanding how this structure forms and how this structure is modified during experience and learning. And why is this important? One reason that this is important is that because it's now recognized that defects in synapse development are associated with a wide variety of neurological, psychiatric, and neurodevelopmental disorders. So here's, again, a cartoon of a normal neuron with a region of its dendrite and dendritic spines, the site of these excitatory synapses. And you can see in, for instance, autism spectrum disorder, this is thought to be reflective in an overabundance of synaptic structures, whereas other disorders such as Rett syndrome, schizophrenia, are thought to be due to a lack of these synaptic structures. And in diseases such as neurodegeneration diseases such as Alzheimer's, it's the synapses that are thought to degenerate first that then lead to the loss of the neurons in these disorders. So there's important mechanisms in terms of understanding human health. So as I mentioned, I'm interested in how synapses form and how they are modified with activity. And the system that we use, as I mentioned, we study the hippocampal synapses, which are important for learning and memory. And we focus on a type of synaptic plasticity, which is called long-term potentiation. And some of you might be familiar with this concept. The idea is that here is, again, a cartoon of a excitatory synapse where you have your neurotransmitter receptors and your synaptic vesicles filled with neurotransmitter that lead to synaptic transmission and nervous system function. But under conditions of activity called long-term potentiation, what happens is that you get recruitment of more AMPA receptors to the postsynaptic side, and this leads to a strengthening of the synapse because the AMPA receptors are important for excitatory synaptic transmission. And in particular, actually here, so it's the recruitment, as I mentioned, of the AMPA-type glutamate receptors that strengthen synapses. So we've been trying to understand how this particular ionotropic receptor is recruited to the postsynaptic structure in response to plasticity mechanisms such as LTP. And so again, this is our cartoon, but we now have a very detailed structure of what this receptor actually looks like. So this is an example taken from the literature of a AMPA-type glutamate receptor that's been a structure that's been solved with cryo-EM. And this is meant to highlight that it's a 4-subunit receptor. So each of these different colors represents a different subunit. So green, red, orange, and blue. So there's it's a tetramer of subunits. And then there's a large transmembrane associated region in which you get flux with the ions. And the glutamate binds to the ligand binding domain, which is shown here. You can see the yellow is the glutamate. And then there's a large amino terminal domain whose function is an area of intense investigation. So we've been trying to understand how this particular receptor, pictured here, how it's recruited and stabilized at synapses in response to plasticity. But it turns out this is quite complicated. So you would think that maybe it's very straightforward. There might be, say, just one pathway in order to get more AMPA receptors to the synapse. But it turns out that these receptors are very dynamic and they cycle between synaptic, extrasynaptic, and intracellular compartments. And this is where my training in receptor trafficking in non-neuronal cells has really been able to facilitate my own research program, because I was studying as a graduate student receptors that trafficked from endosomes to the plasma membrane, again, in non-neuronal cells. But it turns out that many of these mechanisms are conserved whether you're in neurons or in any other cell type within our tissues. And so a lot of the studies, again, like as a student, as a graduate student, as an undergrad and a graduate student, I studied biochemistry, molecular and cell biology. And I never really thought of myself as a neurobiologist, but I'm using all of the tools that I have learned over the years to study this question in neuroscience. So getting back to the AMPA receptors. So this review, this image is taken from a review meant to highlight the different compartments and routes that a single receptor can take at the synapse. So as I mentioned, I told you about that there are the synaptic AMPA receptors that respond to the neurotransmitter from the presynaptic side. And if you wanted to strengthen your synapse, you would need to bring more AMPA receptors into the postsynaptic density. But where do these receptors come from, right? So they can come from a variety of sources. So there are endosomal compartments that store a lot of these AMPA receptors that are necessary for potentiation during plasticity. There are pools of receptor on the plasma membrane, but not at synapses, that are held there in preparation for synaptic plasticity. And there's even receptors that are at the perisynaptic region just outside of the postsynaptic density that can be quickly inserted into the postsynaptic side quite rapidly. So we've been studying aspects of this trafficking. Oh, and then one thing I forgot to highlight is that, so the AMPA receptors are these blue subunits here, and you might have noticed this red auxiliary subunit that's always found with the AMPA receptor. Well, it turns out that AMPA receptors are not just isolated tetramers of subunits. They actually interact with a wide variety of other factors that seem to play a role in all the different sorting and localization steps that are important for synaptic plasticity. And so this is an example. This is a review that I wrote now 10 years ago, demonstrating proteins that have been shown to associate with AMPA receptor complexes and have different functions. And so the classic one that's been identified are these transmembrane AMPA receptor regulatory proteins, which is shown here. These interact with the AMPA receptor subunits and are involved in surface trafficking. They're necessary for the AMPA receptor to get to the surface, as well as anchoring within the postsynaptic density. But there's a large variety of proteins that exist that are unrelated to the original family members. And we've been studying one of these families called the SynDIG family for synapse differentiation-induced gene 1 family. And I highlight this because this was identified using an expression profiling method, functional genomics method, in the early days of the genomic revolution. And so we were able to identify gene products that looked like they were interesting molecules to study because they were expressed specifically in the nervous system, and they were potentially restricted, upregulated during the period of synapse development and plasticity. So I'm not going to go into detail of some of the older studies that we've done to characterize the original family member. I'm going to tell you about our efforts, our most recent efforts. So this is our most recent publication that was published now in 2018. That reflects a combination of research at molecular, cellular, biochemical, physiology, and behavior. And so again, I wanted to highlight the fact that neuroscience is a very cross-disciplinary and collaborative environment. And in particular, UC Davis fosters an area of interaction and collaboration. So I'm going to tell you, give you the highlights of this study, and then tell you a little bit about what our future directions are, and then I will open for questions. So as I mentioned, there's these different pools of receptors. So here's the glutamate AMPA-type receptor that's found extrasynaptic. And we had identified this molecule, SynDIG4, that seems to associate with these extrasynaptic receptors, but not the synaptic receptors. And it turns out that when we looked in our SynDIG4 knockout animal using genetic tricks, these animals were defective in long-term potentiation. And so our model was that, okay, maybe SynDIG4 is important for establishing one of these pools of the receptors that I had mentioned previously. And maybe during LTP, it's this pool of receptors that's important for strengthening the synapses and allowing learning and memory to occur. So I'm going to show you some of the data that were published in this paper. And then I'll tell you about some of the new things that we're doing in the lab right now. So first, we had to generate a knockout mouse in which the SynDIG4 gene, which is also known as proline-rich transmembrane protein, in which we had to remove the SynDIG4 gene in order to try to understand what its function was in the nervous system, right? So one way you can study molecular nervous system development and function is to remove your molecule of interest and then look to see what happens in the neurons in which the protein has been, in which the gene has been removed. So we used homologous recombination to remove the locus, that encodes SynDIG4, and we replaced this with a cassette that expresses the LAC-Z reporter. So some of you might be familiar with LAC-Z. This allows you to have a colorimetric readout of where this particular gene is active. So if we now look at a cross-section of a mouse that has the LAC-Z reporter knocked into where SynDIG4 used to be, we can now look to see where expression of the LAC-Z is as a indication of where the protein is expressed. And as I mentioned, we're particularly interested in the hippocampus, and you can see there's very prominent LAC-Z expression in the hippocampus. And this is the area we've been focusing on for quite some time now. So we can use biochemistry to demonstrate that our molecule is actually removed. So this is a Western blot where we are probing with antibodies against SynDIG4. And in the wild type animal, the ++, we see a robust band. But in the heterozygote or in the knockout, actually this should be the knockout, excuse me, there is a complete loss of the activity. And so this demonstrates that our animals are have lost a SynDIG4 protein. So what's the consequence now of losing this particular molecule? So to do this, we looked at behavior. And as I mentioned, we're interested in hippocampus because it's important for learning and memory. And one advantage of studying the hippocampus is that there's a variety of assays that one can do in order to assess the cognitive ability of your animals. And so what we first did was we did the classic Morris-Water-Maze cognitive behavior test, where mice are placed into a tank of water with a hidden platform that they can't see because the water is opaque. And there are visual cues around the room that the mouse must learn in order to figure out where the hidden platform is, or to strategize where the hidden platform is. So after randomly sampling the entire tank, it will eventually hit the hidden platform, and the mouse will keep swimming because they do not want to be in water. So they will keep swimming in order to try to escape. And so they are able to find the hidden platform over after some point in swimming. So this is before they've learned this task. Now, eventually, they, after more training, so they are put into these tanks on a for 5 days, multiple times a day. And if they're smart, they will start to look around the room and notice that there's cues that they can use in order to remember, oh, the platform, it's by the red triangle. And so after learning, if a mouse has learned, they should be able to orient themselves in the tank to remember that, okay, the platform is away from the green circle and near the red triangle. And you can measure this by calculating the amount of time it takes for the animals to find the hidden platform. So on day 1 of training, wild type animals and mutant animals both take about a minute to find the platform because they're swimming around. But then over 5 days of training, a wild type animal will figure out this task quite quickly and be able to find the platform in about 15 seconds. However, the mutant animal, the one without the SynDIG4 molecule, is never able to really learn this task. There is a slight trend that decreases, but it's as if the animal is always doing this task for the first time. And we can probe this more specifically by doing a contextual memory test, where we now, instead of asking them to find the hidden platform, we remove the hidden platform and we calculate the amount of time that the mouse spends in the target quadrant that used to have the hidden platform. And again, the wild type animals will spend the majority of their time, close to 50%, searching this target quadrant that was supposed to have the platform, and less time in the others. Whereas again, our mutant animal is spending equal amounts of time in all the quadrants. So it really hasn't been able to acquire this learning and memory phenomena. We have continued our efforts. We've also looked at a different type of training where we looked, oh, this looks like it didn't reproduce so well, but we did a different type of cognitive test called the novel object recognition. And so in this case, a mouse is given 2 objects to explore. And on the training day, he's given 2 objects to explore. And then on the testing day, one of those objects is removed and replaced with a novel object. And mice, wild-type mice, will spend more time exploring the novel object compared to the familiar object. And in contrast, our mutant animal has no discrimination between a familiar and novel object. And the reason is because they can't remember that they had seen this object previously, and they're treating these as 2 novel objects. And we don't think there's any discrimination or locomotor activity, because before learning, the mice spend equal amounts of time with both the left and the right objects. Okay, so the behavior is indicating that our mice can't learn. What about the physiology? So we've done, in collaboration with a colleague of mine in the Department of Pharmacology, Johannes Hell, and his postdoc, Lucas Matt, we have done field recordings to measure the activity of these synapses in the hippocampus in response to LTP. And as I mentioned, long-term potentiation recruits AMPA receptors to the postsynaptic side of the synapse. And you can measure that by measuring the field excitatory postsynaptic potential, which is graphed here. So before, this is our baseline, which starts at 100%. And then after the stimulation of long-term potentiation, as I mentioned, you've recruited AMPA receptors, and now synaptic strength has increased. And this persists for at least 50 minutes. In contrast, in our mutant animals, again, without the SynDIG4 molecule, they can no longer potentiate their synapses. You see that in response to potentiation, they actually see no change. If anything, the synapses in these animals start to depress. So we have behavior indicating that they can't learn and encode memories. Physiology, that's demonstrating that their synapses can't be strengthened. So now let's look at the synapses using immunocytochemistry a little more carefully. What's going on at the synapse level, like when we look at the molecules at the synapse level in the absence of SynDIG4? And so this was done by my graduate student, Lindsay Kirk, who graduated now several years ago. And so what she did is she took hippocampal neurons from wild type and mutant animals, and then measured the localization of the AMPA receptor in the wild type and the mutant animals. And she did this by using antibodies against a subunit of the AMPA receptor, GluA1, and a presynaptic marker, vGlut1. And we can define synapses as overlap between the pre and postsynaptic marker, so yellow. And then the extrasynaptic synapses, remember, extrasynaptic AMPA receptors, as I mentioned, there's a large pool of receptors outside of synapses, are areas that are all red because they're not overlapping with green and therefore are not yellow. And then she can quantify the changes in the synaptic versus the extrasynaptic AMPA receptors. And so we were amazed to find that here in the wild type, so in the wild type, you have a large fraction of AMPA receptors at synapses, that's here. And then you also have a fraction of AMPA receptors that are non-synaptic. As I mentioned, there's this large pool of receptors outside the synapse. And in the mutant animals, it turns out that there was a reduction in the pool of receptors that were outside the synapse. But it turns out that what happens is that this led to an increase of this particular subunit at synapses. However, the size of these puncta and the intensity of these puncta were both reduced. And so we interpret this that that there's a change in the synaptic distribution of the GluA1 pool, and because of this, that synapses are now not able to be strengthened. And I think I have a model here. Yes, exactly. So what we hypothesize, and this is our current model, is that SynDIG4, which is this squiggly line in red, is somehow important for establishing this pool of receptors outside the synapse. And during LTP, somehow this pool of receptors gets trafficked to the postsynaptic density in order to strengthen a synapse. And then in the absence of SynDIG4, we're losing aspects of this pool. And so therefore, during LTP, perhaps it can't be recruited to the synapse in order to increase excitatory strength. So this is what we're testing right now. Okay, so I don't want to, and we're going to be using, to test this, we should be able to see, again, using our hippocampal preparation, what's happening in response to LTP. So we can take our hippocampal neurons and then treat them with glycine, which is a way to induce long-term potentiation by within these cultured hippocampal cells. And this project is the thesis work of my current graduate student, Chris Plambeck, who's getting ready to defend his dissertation. And so in response to glycine, we get an increase in, oh, sorry, look at this one. We get an increase in the amount of GluA1 at the surface. Because again, as I mentioned, in response to plasticity, you're increasing surface AMPA receptors. And so you can see the difference between the signal here and here. And then we will look to see what happens, and this is shown here, and then we can look to see what happens in the mutant animal, which is shown here. So we have the wild type in response to the glycine, and we have the mutant in response to glycine. And so here, if you can kind of see, we get this increase in surface GluA1 in response to glycine, indicating that the synapses are being chemically strengthened. But in our knockout animal, again, here and here, it looks mostly the same. And if you quantify it, here's our wild type animals, you get an increase. And in our mutant animals, there's no significant difference between the two conditions. So this is indicating that in SynDIG4 knockout neurons, we're abolishing the ability of AMPA receptors to be trafficked to the synapse in response to LTP. This is our interpretation and our current working model. So the last couple of slides are just examples of some of the things that we're doing now that are ongoing in the laboratory. We're doing a structure function approach to understand what are the domains within the SynDIG4 GluA1 interaction that's important. We're doing super resolution imaging in order to get a better handle of where SynDIG4 is associated with the AMPA receptor at synapses. And then we're also doing live cell imaging of SynDIG4 and GluA1 to see if we can capture this trafficking in real time. So I have a couple of slides demonstrating some of these approaches. So here, this is a continuation of Chris's dissertation project. He's made chimeras between the AMPA receptor subunit and a related glutamate receptor subunit, GluK2. And thus far, he's been able to narrow down the region of the AMPA receptor that's important for interaction with SynDIG4 to the membrane-associated domain and the cytoplasmic domain. So we think that SynDIG4 is binding to the AMPA receptor somewhere along within the membrane of the portion of the cell. This is an example of super resolution imaging with STED microscopy. And this is an ongoing project by Dave Specka, a research scientist in my group. And so here we get a higher resolution. This is a dendritic process and 3 synapses that are found on this stretch of of dendrite. And if we zoom in a little carefully, a little more, so here's the dendrite, here's the GluA1. And if you look at the merge, so the VGlut1 is indicating where the presynaptic input is. So you can see here's the GluA, the AMPA receptor at the postsynaptic density, probably the presynaptic input. And then presumably this is that extrasynaptic, perisynaptic pool of receptor that's within the dendritic spine, but not at the postsynaptic density. And this is what appears to be absent in our SynDIG4 knockout animals. So we think it's this specific pool of receptor that's important. And then finally, we have two new collaborative projects. We're doing a structural modeling approach with another colleague in the Department of Physiology, Yarovoy, to identify a potential structure for SynDIG4. And then we also are collaborating with Madeline Nieves-Cinturon. I should say, so she's a native Puerto Rican who's also in my department of pharmacology. And we are collaborating with her to do live cell imaging of a superecliptic florin tagged GluA1 in order to look at live insertion of the receptor at the surface, which can be measured by the presence of fluorescence in real time. These are just two examples of what we're continuing. Excuse me. And so finally, so we're continuing to test our working model. As I mentioned, I'm just trying to give you a flavor of some of the things that we are doing in the current laboratory. And I've mentioned people throughout the talk who have contributed to the work. This is my current laboratory made up of a variety of students, PhD scientists, and undergraduate students. And of course, because everything is remote now, all of our lab meetings and journal clubs are all done via Zoom. I mentioned our collaborators, past and present funding, current lab members here. And then finally, I just wanted to say thank you to all. In looking back, I realized I was at Puerto Rico just in January 17th, 2020, which seems like ages ago. And I was looking through my pictures and I found this one that it brought a smile to my face because I remember the time spent. It was beautiful weather and it was great food, and it's amazing to think this was only 9 months ago and it seems like forever. So thank you so much. I'm happy to take questions now or at the end of the presentation.
Thank you. Gracias, Elva. Wonderful. I didn't know you were here not so long ago.
I know, it's just like I said, it's amazing. It seems like forever ago, but that was only 9 months.
Mm-hmm. That's right. But you were here like years before as well.
Yes, yeah, exactly.
Visit us quite often.
Maybe almost 20 years.
And also Madeline, that you mentioned, we know her very well because she got her PhD from UPR Río Piedras. And
great.
Yes, and I was part of her thesis committee, so I know.
Oh, no way. Oh, I'll have to tell her that. Yeah.
Dr. Maldonado. I was with Dr. Maldonado, and she will immediately say, "Oh, yes."
Oh, what a small world. Yeah.
It is. So,
mundo pequeño.
So, yes, muy pequeño, eso es así. Any questions from the audience? I think it was really interesting what you were presenting, in particular about the ideas of how the trafficking of these receptors. Jose Enrique, I know you have.
Yeah, so I have two questions, Elva. So the first one has to do, so it looks from your data that the LTP effect has to be like very localized. You have to have the receptors close together and probably in a certain abundance to get a response. So does that sound correct?
For the chemical LTP or Or just the electrophysiology
For
recording?
the electrophysiology, because when the receptors are sort of dispersed, or you have more outside of the synapse, you don't get it?
So it's thought that there's a large pool of receptors that are localized extrasynaptically, and in response to long-term potentiation stimulation, they're recruited exactly very rapidly to the synapse. Now it's thought that those perisynaptic receptors are the ones that are recruited first because they're right there.
Okay.
And then over time, the other receptors that are in intracellular compartments or along the dendritic plasma membrane can then take over and can then come in and further strengthen the synapse for the long term.
Okay.
But yeah, those perisynaptic are thought to be the ones that are recruited initially to strengthen the synapse very rapidly.
My other question has to do with other synapses that don't show the LTP effect. Like, I mean, the glutamatergic synapses are everywhere. Do you see other problems with the animal or other effects that are outside of the hippocampus?
So we haven't tested, there's very strong expression in the cortex, in the cerebral cortex, but we haven't done any electrophysiology of cortical neurons. That's something that we could very straightforwardly do. Well, I should say my collaborators, because I'm not an electrophysiologist. And yeah, so I don't think we've looked, it's not expressed in the cerebellum at all. So there's types of plasticity that exist in the cerebellum, but since it's not expressed there, we can't test that, or, we could, and it should be negative. No changes, I mean. But yeah, so we haven't tested anything outside the hippocampus. But the mice themselves, again, they seem fine. They're able to breed and reproduce. We get normal Mendelian levels. It's just that they can't learn. They can't remember.
And have you looked at any perhaps any rewarding behavior like food reinforcement or?
No, we haven't, and that would be another. There are physiology as well as behavioral tasks that are also hippocampal dependent, but also amygdala dependent. And so we could tease out whether it's strictly a hippocampal important phenomena, or is it also involved in other types of brain areas.
Okay. And do you see this phenomenon also in long-term depression as well? Or?
So that's interesting. So long-term depression, and I didn't go into detail too much. So let me just share just the one more thing, because I do want to here. So long-term depression. What I didn't say. So these are the receptors that we think are immediately recruited to the postsynaptic side in response to long-term potentiation. But it turns out in response to LTD, there's also a pool of receptors that are initially recruited to the postsynaptic density, and somehow this leads to a alteration in the postsynaptic density and ultimately loss of receptors. So there's two pools that are potentially at play here. And when LTD was looked at in the SynDIG4 knockout animals, it is also deficient. I don't have that data in this presentation, but it turns out that that initial recruitment of receptors to the postsynaptic density in response to LTD, it's thought to remodel the postsynaptic density in such a way that you can then get permanent removal of receptors to get long-term depression. And that's also deficient in SynDIG4 mice. So it might not just be the recruitment, but it might be somehow the remodeling of the postsynaptic density in order to get long-lasting changes.
Very interesting. Any other questions? Oh, we have a question.
Yeah.
The behavioral effects of the knockouts could also affect fear memory? For example, maybe knockout mice were never learned conditioned fear response.
Yeah, that is something we definitely need to do, Hector. We need to do the fear memory, because that is both hippocampal and amygdala dependent, and that would help us to understand, is it solely hippocampal, or is it a general phenomenon? So that's something, again, I'll need to find a collaborator, but there are collaborators here at Davis who do that, so we could easily test that.
Okay, great. Wonderful. Okay, any other questions before we move to the other as well important part of our seminar? Great. Okay, so now we're going to have Dr. Martin Usrey to talk about the graduate program in neuroscience at UC Davis Medical School. So I'm gonna go and get your presentation. And I thought it was here, but I don't see it.
Great. And while you're doing that, you can hear me just fine?
Yes, we do.
Okay.
Hold on. I thought I had it. Yes, I just have to open it again. Okay. Apologize for that. Everyone can see it? Everyone can see that?
Great. Well,
yes, we can see it.
Wonderful. So, Marty, take it.
I will. Now, folks, before we begin, I want everyone to unmute themselves, because you all deserve a round of applause for picking the most interesting subject in biology or in science to study. So please undo your microphones and congratulate each other. Awesome. All right. Well, thank you so much. So yes, it's a real pleasure for me to get to tell you about what's going on at UC Davis in terms of our admissions, our graduate program, and the diversity efforts that we have in place. In addition to myself for this portion of the talk, Eden Berrigan, a graduate student in our program, is also joining us, along with Ashley Hodell, our academic coordinator, to help answer questions at the end. So let's go ahead and Dr. Maldonado is going to advance the slides for me, so I'll just say, go ahead and let's advance. Perfect. So where is Davis? All right. It is in California. This map here, oh, and let me just take a moment to tell you, I put more information on these slides than I'll actually state, just because I know many of you will go back and look at them afterwards, and there's some information in here that you might find useful. So the city of Davis, where UC Davis is located, it's just west of, and Dr. Maldonado, it'd be great if you used your pointer to point out the items I'm talking about.
Okay.
So yeah, so we're just west of Sacramento, where that pin is in the map, and just a bit east of San Francisco, about 75 miles east of San Francisco. And between us and San Francisco, we also have the wineries of Sonoma, Napa Valley. To our east, you might notice that blue lake, that's Lake Tahoe. And all around there are lots of ski resorts. So surrounding UC Davis, we have all sorts of activities to enjoy that are listed below: swimming, sailing, hiking, my favorite, skiing. So quite a few things to do. Also, our students love to cook and to dance, and it's just countless opportunities for things to do. All right, let's go on. So the community of Davis. Now, Davis is a classic college town. What that means is that the city revolves around the college. And there's a handful of college towns in the United States, and Davis is one of these. If you look at Davis, I put down some interesting facts about the community. So we have the highest number of biologists per capita in the country in Davis. We also have the highest number of bicycles per capita in the country in Davis. In fact, the League of American Bicyclists named Davis the most bike-friendly town in the United States. So what do we have? Oh, and then the next thing, the Today Show, many of you have probably heard about that, has called us one of America's 5 friendliest cities. So you put all this together, and what are we? We like biology, we ride our bikes, and we're nice people. So it's really, it's a great place to live, and it's a great place to be. Again, Outside Magazine thought we were one of the 10 best towns to live in the nation. And another interesting fact, because we are a college town, our residents, the folks that live in Davis, have the highest level of college graduates amongst those residents in the state of California. Okay, next slide. Now, the university that's at Davis, it has a little over 30,000 students. Now, keep in mind, our town's population is about 65,000. So a third of the people that live in our community are students. Amongst that 30,000, a little over 6,000 are students, are graduate students or professional program students across 87 different programs. We are the largest campus in the UC system, over 5,000 acres. And there's a few other facts here that we're pretty proud of. Forbes named UC Davis as the #1 best value school for women in STEM. We're also ranked #1 in the country for graduates, for female and underrepresented graduates in the STEM disciplines. And then, believe it or not, 1 out of every 267 Californians is an alumnus of Davis. They've come through UC Davis and been part of one of our programs. Okay, next slide. Now, that's our town. What about our graduate program? Well, we're rather large. We have over 90 faculty members, and their research interests span all the way from molecules to perception. So Elva, as you just heard, studies molecular mechanisms of synaptic plasticity. In my lab, I'm at the other end of the spectrum. I use awake-trained monkeys that play video games, and I record the neural activity in their brains while they're playing these video games to study neural mechanisms of attention as it relates to vision. In our program, we have about 50 doctoral students, and we take care of these students very well, as you'll see from the benefits below. You might not know this, and you should. When you're a graduate student, you get paid. So our stipend for being a student right now is over $34,000 a year. And that's to allow you to focus on your studies and on your research to be successful. This financial support is provided to every student who's admitted to the program and throughout their stay in the program. We also, for our first-year students, before they've joined a lab, they receive funding to attend a national conference. It's really important to get out there and to see what's happening in the national arena. So we send our students to those, and of course we also include medical benefits, health, vision to all of our students. Okay, next slide. Some other facts about our program. Our faculty, there's about 90, as I mentioned. We aren't a departmental program, so the people that participate in our program don't have to do it. They want to do it. So our 90 members are those that apply to be a member, to be a mentor, to participate in our program. So they're committed to the program that we offer. We have just a vast array of technologies available to our students, far more than I could go over right now. And with the infrastructure that we have and the dedication of our faculty, our training record turns out to be really strong. Over 90% of our trainees go on to careers in academia or research. And in fact, in our most recent review from NIH, they commented that the scholarly progress of our students is impressive. Okay, next slide. Now, our curriculum. When you come to Davis, our goal is to teach you broadly how to be a neuroscientist. We want you to have an understanding of neuroscience from molecules to cognition and perception. So our curriculum reflects this. Right now, our students that have just arrived have been, are starting a boot camp. And this is a crash course that begins before formal courses starts. Then you can see our core courses that go from cellular to molecular, systems, cognitive, anatomy. And this is to give our students that foundation that they need. And our goal in the end is that a student who graduates from our program should be able to go to a conference as large as the Society for Neuroscience conference, walk into any room, sit down where a talk is going on, and have the basic knowledge that they need to understand what's happening. And that's our goal. We also require journal clubs of our students. The first-year students do laboratory rotations to identify the labs they're going to join. They do a TA-ship to get some practice with teaching. And then, as with other programs, you have preliminary exams, the thesis proposal, your written dissertation, and your exit seminar. And all of this then takes place over the course of about 5.5 years. Okay, next slide. Now, we have many, many other courses available. I've only put a few of these on here, and the 3 that I want to highlight are those that emphasize career development, because that is something we really stress. We want our students that when they leave to have the skill set that they need to be successful. So the seminar speaking course, a career pathways course, that is how do you take that next step, how do you land your next position, and also a grant writing course so that you're learning how to be successful in writing grants, because that's so important for what we do in the field. Okay, next slide. Okay, now within our program, we are big. We have lots of seminars, research seminars taking place. On a weekly basis, there are more seminars than you could possibly attend. I've highlighted 3 at the top here that I want to just tell you about. The first is a graduate student seminar series. This is a series that's run entirely by the graduate students. They pick the speakers, they organize the visit, they host the individual, and it's really a wonderful event. There's 3 of these per year. Below that is listed our SOMA seminar series. This is the Seminar Outreach for Minority Advocacy. As you'll see as I move a little further along, promoting diversity in neuroscience is a strong, strong emphasis of our program. And we appreciate this, and we want to learn more about it. And we run a seminar series so that we can, in fact, learn more about this. And the SOMA series is one of these, where the graduate students, again, organize this, bring in outside speakers, so we can learn more about the research that's taking place by these individuals, but also how to be more aware of the issues surrounding improving diversity within neuroscience. And then lastly, the one I have on there is a Perspectives in Neuroscience series. This is offered through the Center for Neuroscience in our program. And this is, I mentioned this one, this happens about every 3 weeks. And this is one where the students meet with the speaker at each and every one of these to have lunch and to have more of a one-on-one conversation with the speaker that's come in to talk to us. And then several other seminar series are listed below. Okay, next slide. Okay, now we have a retreat. Many programs do. It's one of the most fun things that we do every year. It takes place over 2 days. It's run by the students. We rotate between 3 just wonderful places to go: Lake Tahoe, Point Reyes, that's on the coast, as well as Bodega Bay. It's a research lab of UC Davis that's also on the coast. It includes a mixture of student talks with faculty talks. They're all short so that lots of people can present. We want to learn as much as possible about what's happening across our broad community. We invite an outside speaker to come in, our keynote speaker, to also present and meet with everyone more closely. And then at the bottom there, I've mentioned that it's really, the goal is to have a mixture of science along with social activities, because it's a big purpose of this is community building. And if you look up at that montage above, the lower left panel in that montage, there's a few goofy looking individuals. You'll see Elva to the right of me. I'm wearing the red, and that's Elva in the white lab coat with the spiky hat. And really, you know, there's skits, there's all kinds of fun things that we do to have a great time at this retreat. Okay, next slide. Now, let me tell you a little bit about how to apply to the graduate program at Davis. There's an online application process. The deadline to apply is December 1st. There's a link for doing this. It'll be open just a little bit later this month, and I've provided the information for that link there as well. Excuse me. It's a pretty typical application in that we need transcripts from each of your college level institutions, 3 letters of recommendation, a statement of purpose. And again, something that we really truly care about at Davis, we want a personal history and diversity statement. That's something that we take very, very seriously and weigh very strongly. The GRE is not required to apply to our program, and we do have a fee waiver program. Okay, next slide. Now, let me take a few minutes to tell you about some of our outreach activities and the programs that we have to promote diversity in neuroscience. And this is, you know, I could go on and on about this because there's so many. The first is we are a recipient. Some of you in the room will know what this is. It's the NINDS Neuroscience Development for Advancing Careers in a Diverse Research Workforce program. This is a program that funds undergraduates to be in the research labs and also enhances their academic experience to increase the flow of the pipeline to bring them into graduate school in neuroscience. I mention this because our graduate students also participate in this program. They can serve as mentors to the undergraduate students, and in fact will be their buddy when they go and present their own work at, say, the Society for Neuroscience meeting. Below that, I've mentioned the SOMA seminar series also, but also what we have is a weekly reading group or a journal club where our students get together and read scholarly articles about how to promote diversity in science and the issues and obstacles that need to be overcome for that. Down below, I have another picture montage. This is from Brain Awareness Week. And now this is when we go out to our community and try to engage the community to get them excited about brain research and neuroscience. We go to the farmers market. We hold events at our research seminar, at our research centers. We also go to the local schools and give programs to the kindergarten through 12th graders for this. And again, the goal is to increase that flow in the pipeline to bring those individuals into college and then ultimately into graduate school for neuroscience. Next slide, please. Okay, and this is the last program that I want to tell you about, and I'm hoping this is a program that will excite many of you that are here and that you will apply to this also. This is the Neuroscience Initiative to Enhance Diversity at UC Davis. This is a program that we seek students from all over outside of UC Davis. It's a 2-day, all expense paid workshop. So we pay for your travel, we pay for your room and board for this. And it's for undergraduates that are interested in a PhD in neuroscience. And some pictures down below just show some of our past NEED events, the students that have come in for these, and some of the activities that we have that take place. If we go to the next slide, I have a list of the things that the students do. So these are the workshops that we run. And the purpose of this, let me step back. The purpose of this is to take students that maybe aren't so sure about whether neuroscience for a PhD is something they want. Maybe they want to learn more about it, or maybe there's students that they know this is what they want, but they want to make sure they can make themselves as competitive as possible. So we run all the workshops listed over on the left. What is graduate school? We do have something for what are the pathways to medicine for those that are on the fence about PhD or MD. We talk about graduate school from the perspective of students, how the admissions process works, how to be successful in research. And then some very, very important workshops, how to prepare your best application, acing the interview. Once you've been invited to interview for a program, yes, you're going to be doing a whole day's worth of one-on-one or small group interviews. So we teach you how to interview, and we do it sort of like speed dating, where you interview, and our own graduate students are the interviewers. You interview, you get feedback, you rotate, have another interview, hopefully incorporate the suggestions that you got, and then rotate and do another one. And then we also have a discussion about career journeys, and for the faculty that we have that have made the different pathways that they've taken to get to where they are. And the take-home message from that is that there is no singular track. Everybody has a unique way that they've navigated to get to where they are in their career in neuroscience. Another key thing, and I bring this up because it was a discussion last week that I heard, we have a big focus on imposter syndrome. That is something that everybody experiences all the way through. And we have a whole session on that to break down the barriers of imposter syndrome and to teach people that it's something everyone has. There's nothing wrong with you if you feel it. And we have a really nice conversation about that. We do lab tours, campus tours, data blitzes, poster sessions, and then we have a lot of social activities as well. So this is a program that we track. We want to follow this very carefully. We've had well over 100 students from across the country, and we've had several from Puerto Rico come. Over 50% of the students that come, we track them, have gone on to PhD programs or other professional degree programs. So we're really proud of the fact that that I don't know if it's our event that helps shape them and motivate them, but we do stay with the students afterwards. We have our students that are in the program serve as mentors that are available after the students leave to help them as they're preparing their applications, or just to talk to them about, you know, how things are going. Okay, so last slide. So this is a flyer for our program. We can email it, a separate flyer, to everybody that's interested, but I did want to tell you how to apply. It's a little bit hard to read on this slide. The dates for the next NEED event will be in April. It'll be held on April 22nd and 23rd. The deadline to apply is January 31st. And again, we bring in about 25 students each year for this. The link for applying is down below there, so you can apply online. And then lastly, if you have any questions, you can contact Ashley Hodell, who's also part of the meeting right now and can help answer questions about the NEED event, along with Eden, who I see smiling there as well. So anyways, that's in a nutshell, neuroscience at UC Davis. We greatly value our students. We think we offer them a wonderful experience, and we truly believe that we set them up for success once they graduate from our program. So why don't we stop, and the three of us, along with Elva, would be happy to answer any questions that you might have.
Great. Questions? Okay, already we have some. There's a question. What are some of the things you make for a good? What does it make a good application for the NIED?
Yeah, great question. Eden, have you been on the selection committee?
Yes, so I was actually on it last year, and I'll be on it again this year. The things that we really look at are personal statement, so kind of your story, what you're interested about. Also, we look at letters of rec. So a really strong letter of rec, even if you haven't been in a lab for a long time, or even if it's only from a professor that you took a class with, if they can say something about your character or things that would indicate that you have a lot of potential to be a really good scientist, like you're resilient, you're very curious, like those go a very long way when we're looking at the applications. And we also like to see younger students, although if you are a senior student thinking about a post-bac, this is also a really good event to come to and learn more about other options.
Okay. You guys can unmute yourselves because we're a small group. So any questions for Eden or Marty or for Ashley?
Yeah, so I was wondering, does UC Davis offer any type of prep or post-bac program?
Yeah, I just put that in the chat. So there's a prep at UC Davis. This isn't neuroscience specific, but I have hosted a prep scholar in my own laboratory. I'll pull up the website because the application for that, I think, is also sometime in the spring. But I'll pull that up and put that in the chat.
Okay, thank you.
There is a student, yeah, who's currently in it. If you would like me to connect you with her, who is a neuroscience student in the prep program, Maribel, if that would be helpful.
Yes, that would be
good
awesome.
idea, yeah.
Yeah, I can send you my email in the chat. Anyone feel free to reach out. I have nothing but time to answer emails.
Thank you so much.
Of course.
Any other questions?
Hello. I might have missed this, but is there a teaching requirement for PhD students at UC Davis?
We do have a teaching requirement. It's 1/4 of TAing. So, you know, if you want to teach more, you certainly can, but all of our students need to do at least one course.
Awesome, thank you.
Sure. And I have to say, it's really fun. You know, getting out there and being part of teaching the undergraduates, it's an exhilarating experience.
Okay. Any other questions?
I see Ashley's face now. Hi, Ashley.
Hi.
How many PhD students do you guys accept per year?
That number ranges from between about 8 to 13. And of those, about a third of the, so about 8 to 13 per year. And our program is a little bit less than 50, and a bit over a third of our students come from underrepresented backgrounds.
Okay,
so it's a smaller class size, but we're also growing. So we are trying to expand the numbers so that we have larger class size going forward. We want to expand our program.
Okay, so you said that the coursework lasts for about 5.5 years?
No, the time to degree takes about 5.5 years. The coursework is largely done after the first year, and then you're spending your time almost 100% in the laboratory doing your research.
Yeah, because I wrote it down and I was a little confused about it, and I wasn't sure if I had heard something else or not.
Well, I'm really glad you asked. Yeah, I'm really glad you asked, because no, that would be quite something to have to take courses for 5.5 years.
Yeah.
Hello, I have one more question, and I'm sorry. Okay, so is the TOEFL a requirement for upcoming PhD applicants? And if so, what are the approximate required scores for Puerto Rican students or for just general TOEFL taking students?
Elva, do you want to field that one, or I can?
I think you better. I'm not actually sure.
Right. So the TOEFL is only required for international students. And I don't think there's, maybe there are some international students online, but I sort of doubt it. Puerto Rico is not international.
But for some institutions, even though you're correct, we're part of the US, but their argument for asking a Puerto Rican student for the TOEFL is that they did not have their education in English. So the argument is that there's a concern about proficiency of the language, and some schools bring it up, and then they also have a cutoff on the score you're supposed to have in order for them to even consider, or maybe for you to have to take the TOEFL again. This is a matter that, if you ask me, I think is something that for our students it's not necessary. I think it sends a wrong message. And but you know, I'm not in those institutions, so I cannot persuade them for them to take it out. But yes, that's why Amanda is asking, because in some we have our students have a table where they have columns of requirements, and so they need to know, because if that's the case, they need to take it prior, obviously, to submit your application. And the fact that you don't have GRE, which is wonderful as well, I congratulate you for taking that out. But now then they ask you for the TOEFL, and then that doesn't make any sense to me, because if you're taking out the actual, you know, exam that measures, you know, your knowledge in biology, but then you have to ask them, okay, do you know how to speak English? It doesn't make any sense to me. But you know.
I will check, but I didn't think we required TOEFL for except for international students. But I will confirm. So I would not expect anyone from Puerto Rico to be required to have a TOEFL.
I found online, I don't know if our department is different, but online it says that UC Davis Grad Studies requires it for applicants who either did not receive a prior degree from an institution where English is the sole language of instruction, and or are not from a country or region where English is the language of instruction. But again, I don't know if neuroscience has their own set of rules, but that's the UC Davis grad study rules.
Right, but I think, but wouldn't the Puerto Rico fall under the non-international? No?
Yeah, but the first one.
Yeah, the way they're listing it is as based on the language of instruction or the main language of the country. So it's actually, the way it's worded from grad studies is actually not about nationality.
Sure, you can be in Peru or Mexico and go to an institution that is solely in English, but you have to prove it. You have to show everything, and that's what that is all what about. And I understand the reason, but
I do. Yeah, most schools have asked me about it just solely on the fact that I'm not from an English-speaking institution, so that's why I emphasize it.
Right. Now, that's a good question to ask, and that's something that I should look into, and especially now as we've just recently gotten rid of the GRE, it seems like it might be a good time to also revise the institutional requirements here. So that I'll also look in to see what at the institutional level the conversation is going on. But
and also it would be valuable for the students to know the cutoff, because like I said, some of them ask at least a minimum of 20.
Yeah, Eden put in the chat. I guess 80. This was from grad studies.
Mm-hmm. I can post the link if that's helpful.
80. You're asking
so you want to fact check me.
It's good.
And there was another question about summer programs geared towards neuroscience. I think that would be the R25, right, Marty?
We have a STAR program that takes students from everywhere. It's in the College of Biological Sciences. It's not restricted just to neuroscience. It's in anything in biological science. So we do have a summer program for that. And then we also have a pending application for an R25 with the University of Nevada, Reno, to do a summer program that would take students more broadly as well. So we do have one program in place, the STAR program. So if you were to Google that, you would find it.
Great. Any other questions? Oh, Marty.
Yeah, I have a question about the NIED application program. And one part, it says about my ethnicity or race. I'm both Puerto Rican and Mexican. So I want to know if I need to put both, check out the both of them, like Caribbean and Mexican, or just one.
Great question. You know, the application is one where you can put down all of the information that you want to share. And in addition to the items that Eden mentioned, I mean, we also are very interested in, because what we're really trying to do is get information out to folks that may have some difficult time getting it from where they're located. So if there are some obstacles that you've encountered, those are other things that we also want to hear about.
Okay, thank you.
And there's a question about other academic, do you accept students from other academic backgrounds? Is there a list of required courses? I'm assuming for applying to the Neuroscience Graduate Program? Yeah, exactly.
Yes.
Yeah, so we accept students from all different backgrounds. Many institutions don't have a neuroscience or neurobiology undergraduate degree. So you might be very interested in neuroscience, but just not have had a opportunity to major in it, for example. Maybe you've taken some courses, you know, like neurophysiology or psychology, so you have some background, but you really know you want to do neuroscience. So then you would focus your application on what it is about neuroscience that drives you for the PhD. And this is also why we have the very rigorous core curriculum at the first year, where we cover all the components of neuroscience. Remind me, Eden, what was your undergrad degree? Were you neuroscience from Irvine?
I was biology.
Biology, yeah, exactly.
And a lot of students also come from psychology.
Yeah.
So like you also can get in if you don't have a BS necessarily, because a lot of institutions only give out a BA in psychology, so you're not limited by that either.
That was going to be my next question, because my bachelor is not a BS, it's a BA. So yeah, well, thank you. Is there a list of required courses though? I mean, like chemistry or all that stuff that you need to, so I can, because I know that sometimes when you apply from graduate school from background that's not in science, they require you to show that you can have a basic knowledge. So
yeah, so I would recommend, if your background, if you have like a small number of science courses, one concern would be, would you be prepared for the rigor of the graduate curriculum? But you can demonstrate this by saying, oh, I audited a course in molecular biology, or I audited a course in neurobiology before starting grad school, before starting your application, in order to prepare yourself. And this will also demonstrate to the admissions committee that you're really interested in pursuing this degree, right? Because you recognize that your limited background, for example, and you've at least done auditing or taken a few classes like at a community college level. But there isn't a list of required courses. It would be something the admissions committee would want to ensure that the applicant would be prepared for the rigors of the graduate curriculum.
yeah, sure. I mean, it's just that I did some research, undergraduate research, and it was in neurology and everything, but my bachelor is not on science. I'm a teacher, so I just wanted to know, because I'm really interested and I fell in love with it. But
Great. Wonderful. Any other questions?
And I was just going to highlight one more thing. I don't know if Marty mentioned, so we use a holistic review process for admissions. And so we look at the whole candidate rather than, some minimum GPA or some minimum score. It's really a holistic review process during the admissions cycle.
great. Thank you so much.
You should apply.
I am strongly considering it. So yeah. Thank you so much.
This is great.
Great. And Ashley, why don't you say something, because I want people to see your face for the as the contact person for NEED.
Sure. So I'm the academic coordinator with the Center for Neuroscience. I administer 2 of the training grants offered there at the center. And I also am the coordinator for the NEED event, working with students like Eden, who's been fantastic to get that event going. So I am happy to be your point of contact for any questions about the programs offered at the Center for Neuroscience, and I can help field those to the appropriate people if I can't answer your question.
Great. Yeah, Eden just mentioned she's the student rep on the admissions committee as well. So she's a really good source of information for many of the opportunities at UC Davis Neuroscience.
She is the student rep for everything.
I've been here a while, so also if you're curious about labs or questions about that, I got it.
Can you share your email in the chat, Ellen?
Yes, but I can put it again. It might have gotten buried. Here you go. There you go.
Gracias, Ellen.
For anyone else. Of course.
Español, Ellen.
Yep, that's I get that then sometimes,
You get that, ¿verdad? I imagine. Well, I think we're 6:30, time to say goodbye because we want to get everybody a chance, and to also really thank you all for being here. It's a wonderful opportunity for our students and for myself to be part of this partnership, because I think this is a partnership of us from our students and from institutions such as UC Davis that is so committed to our community. So I really appreciate you being here. And I encourage you, I know you're all busy, but to come back next week, because this is the whole semester, every Wednesday at 5, where this next week we'll have Dr. Carlos Eisman from Brown University, and he will be talking about the role of matrix melatoproteosin 9 in neurodevelopment and neurodevelopmental disorders. So please join me again. So thank you, Marty, Elva, Ashley, Eden. Thank you so much for being here. I really appreciate it. I'll see you guys.